Translation. The ribosome binds to the mRNA at the start codon AUG that is recognized only by the initiated tRNA. The ribosome proceeds to the elongation phase of protein synthesis. During this stage, complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. The ribosome moves from codon to codon along the mRNA. Amino acids are added one by one, translated to polypeptide sequences dictated by DNA and represented by mRNA. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete peptide from the ribosome. Tay-Sachs disease. Tay-Sachs disease is a rare inherited disorder that progressively destroys the brain and spinal cord nerve cells. The most common form is found in infants, where they lose motor skills such as sitting, turning around, crawling. This later progresses and the child is less able to perform movements due to weak muscles, mental illness and speech difficulties. This disease occurs about 3 to 6 months, and most children do not survive after 2 or 3 years of age. The hexa-A gene, which is mutated, is the cause of this disease. It is a gene that instructs part of an enzyme called beta-hexoaminidase A, important for brain and spinal cord functioning. This enzyme is located in the lysosome that breaks down toxic substances and acts as a vacuum center. Beta-hexoaminidase A in lysosome breaks down a fatty substance called GM2 ganglioside. Mutations in the gene affect the activity of the enzyme, and GM2 ganglioside cannot be broken down. Therefore, it accumulates to toxic levels and destroys the neurons in the brain and spinal fluid, causing Tay-Sachs disease. Inhibition of endoplasmic reticulum associated degradation versus native folding and loss of function protein folding diseases was found. This article was found in the Journal of Biological Chemistry and was published on October 17, 2011, very recently. In this study, they have exploited a process called endoplasmic reticulum degradation, which is the mechanism by which only corrected folded proteins are transported to the final destination, and misfolded proteins are degraded. It is a selection process. They have achieved to inhibit this process by cell treatment with small molecules that interfere with recognition or retrotranslocation of misfolded substrates. Overall, they managed to correct the activity of this misfolding, but have found that ERAD inhibition is indirectly proportional to the loss of lysosomal activity, and that it produced varied levels of stress, unfolded protein response, and induced apoptosis in the endoplasmic reticulum. Therefore, they were unable to combat this problem. Listening to our presentation, these are the references where we obtained all our work from. I hope you enjoyed the presentation, and thank you very much once again. Goodbye.